Hello everyone, welcome to my YouTube channel. Today we're going to be looking at something that I think will be very beneficial for you guys. So I had already made a video on the working principle of full blood count analyzers. So where we look at something like impedance, where we look at something like aperture and so on. Also we look at something like scatter plot by flow cytometry and all of that. We also look at maybe the different parts of full blood count analyzer, whether 3-part or 5-part or 6-part, 7-part. I'd already explained all of that. But I want to say that those principles that we've mentioned has to do with how the analyzer get the values, okay? But for the analyzer to get the values, a lot of things happen. Remember that this analyzer, they make use of reagent, and those reagents, they do a lot of things before the whatever thing that is being measured, before the quantification, value that you get, following the flow cytometry or impedance, based on aperture, as the case may be. So today we're going to be looking at the working principle of the analyzer reagent, and I'm going to be focusing on Sysmex XN. So I'm going to be focusing on Sysmex XN line. So when we talk about Sysmex XN line, we're looking at a line. So that word, the key word there is a line. So when we talk about the line, there are different equipment you're going to see. Number one, you are going to see the track where you can load the samples. So from the track where you load the sample, the barcode of the rack and also the sample will be scanned. Then it moves across to start entering into the analyzer. So that will be the maybe Sysmex XN 9000, Sysmex XN 9010, as the case may be. There are different code for it. So it will then move to depending on how many analyzers that the hospital have. Some people might have 3, some people have 2, some people have 4, some people have 5. So the longer the more the analyzer, the longer the line will be. So this rack will start moving into one analyzer to another analyzer and all of that. Anyway, once the full blood count is completed, what will then happen is this, and there's nothing else to do. It will move from that to what we call tube sorter, okay? But sometimes it may require to make a film. Maybe the analyzer thinks from the result of the patient, it wants to make a blood film. So what would then happen? That rack will move out of the full blood count analyzer and back to the line, and then enter into another analyzer, which is what we call SP10. So far I've mentioned 2. I've mentioned Sysmex XN, maybe like 9000, 9010, and so on. Now I've mentioned something like SP10. So this SP10 is the one that we use for making a blood film if the analyzer thinks that it requires blood film to be made. So what would then happen is that once the blood film is made, once it's finished that, then it will again come, the rack will come out. Once the rack come out, it can then be back to the line. Then it can then be transported, transported to the tube sorter. So the tube sorter again would then scan the barcode of each of the samples on that very analyzer. That is where under the tube sorter, that is what we call maybe TS, maybe 500 and so on. Okay, but it generally is called tube sorter. So that tube sorter would then sort the samples, sorting in the sense that has it finished, is anything still outstanding, is it anything that is unknown, and also so anything that has completed, it will just archive it. So when we talk about archive, it basically means that everything has been completed, everything is fine. So it will just archive it. Then from there, we can decide, is there any sample that need to go for ESR? So the one that need to go for ESR, we then put into the ESR rack, which we call buffer rack. So once you get into the ESR rack, then from the ESR rack, whenever it is due, is ready, to move into the ESR analyzer, then another rack will come. Then it will pick it. If it to pick the sample, then the tube sorter will then carry the sample, put on the rack, then take it to the ESR analyzer, and that is what we call interliner. So when we talk about Sysmex XN line, this is what we're talking about. You're looking at from the track, okay? Then you look at loading the sample from the track. Then you look at the main analyzer of the full blood count, which is Sysmex XN, maybe 9000, 9010, and so on. There are different code for it. Then the next analyzer you see by the side is the SP10, which is the one that make the blood film automatically. Once the analyzer thinks it wants to make a blood film. Now after that, you have another equipment, which is a tube sorter, okay? So like TS500. Then the tube sorter will sort it in the sense that, okay, I have finished this, or maybe something is still outstanding, or maybe there's still something that I don't know, as the case may be. It will just sort it out. Whatever you see the sample will give you an idea of what the problem might be. Has it finished? Does it require ESR, or maybe is there anything outstanding? The one that requires ESR will then later move to the ESR analyzer. So when you talk about Sysmex XN line, you're looking at full blood count analyzer, blood film making analyzer. They now look at tube sorter. They look at the ESR analyzer. So the whole thing are connected with line. So that is why we call it Sysmex XN line. Okay, now I know I've talked too much before telling you guys my name. Once again, my name is Dr. Emmanuel Ogbodo. I've been working in the UK as a specialty biomedical scientist. I'm a lecturer here in the United Kingdom in biomedical science department. So yeah, let's get into it. So what I've decided is that I'm going to pick this one after the other. So I'm going to first of all talk about the Sysmex XN 9000 or 9010 as the case may be. So I'm going to be looking at the CBC full blood count analyzer. Then after that, we can look at the SP10. Then after that, we can look at the tube sorter. Then after that, we can look at the ESR. Okay, so I hope to cover all of that for you. Okay, now let's get into it. When we look at full blood count, which I've just mentioned is going to be Sysmex XN 9000, 9010. There are so many codes for it, okay? So now when we look at it, what happened that there are things that I want you to know, but I think I really want to start off by saying this. There are different channels, or there are different things that you measure. Remember that in full blood count, you're looking at blood cells generally. You're looking at the red blood cell, you're looking at the white blood cells, you're looking at the platelets. Okay, even in the when you talk about the white cells, you're looking at total white cell count, then you look at the differential white cell count. So we can look at this like WBC, which is white blood cell, WBC differential. You look at reticulocytes, which is retics. You look at platelets, or maybe Platelet-F, okay? You look at WPC, okay? So what it means that white cell count differential, retics, Platelet-F, I'm going to explain that in detail, Platelet-F, and also something like WPC, they are all measured fluorescent flow cytometry. I'm going to take that again. WBC differential, retics, Platelet-F, okay? Platelet-F and WPC, okay? They are all measured with fluorescent flow cytometry. When you look at something like red blood cells and platelets, they are measured with hydrodynamically focused impedance. So that's how those ones are measured. Then we look at the hemoglobin. The hemoglobin, of course, is then measured with, for you to measure it, you're now looking at something like cyanide, where you have cyanide, free sodium lauryl sulfate. That is how the hemoglobin is measured. Then, in saying that there are different channels that when it comes to measuring full blood count, there's a lot of parameters like you already know. So each of these parameters, they can be measured in different channels. So we'll have a lot of channels. So I'm going to try to list them. So we'll have something like retic channel, WBC channel, platelet channel. Then of course, under the platelet, you can also have Platelet-F. I told you guys I'm going to explain that Platelet-F further because I think it's very, very important. So sometimes when we do a full blood count, you might get a platelet count, and that platelet count may not actually be the, how will I put it? It may not be the actual platelet count of this patient. Remember, there's some of the platelet that may be immature, okay, or something like that. So, but if you really want to get more functional platelet count of a patient, you might need to do what we call Platelet-F. What the Platelet-F does basically is that following the lysis of the blood, okay, so nothing will happen to the platelet. The platelet will still remain intact after the lysis of the whole blood. The platelet will still remain intact. But what will then happen is that with the help of the fluorocell Platelet-F, okay, that will then extend the RNA of the platelet. That will make it more specific, and that is then what that can be measured. And in measuring that, because could be other cells that may be of the same size of the platelet, or something like fragment that could possibly be interfering with that. But because of that RNA has been stemmed, it will then help the analyzer to measure the actual functional platelet count of that very patient. So that is where Platelet-F comes in. So in other words, Platelet-F is more accurate than when you just perform normal platelet count, okay? Because with the Platelet-F is more specific, because the fluorescent Platelet-F will then stem the RNA of the platelet, okay? And that will now help the analyzer to measure specifically about the platelets, okay? Now, let us look at some other reagents, okay? So I thought I should start off with that. That may be very, very vital. So, but I'm going to try to start by telling you, I did mention it actually when I was talking about the principle of analyzer. But let me also repeat that. So when they use their reagent, there are two main reagents that they use. And I'm going to generalize it. And once you think about it like that, it is easier to understand the principle. In other words, when you go for interview, even if it might look like you may not be able to remember everything, once you understand this, you can have a way to generalize the answer in such a way that the interviewer will also be happy with your answer. Okay? Now, let's look at it this way. So when you look at the Sysmex XN full blood count, you're looking at a few things. Number one, what you are looking at, number one thing is the Lyser cell reagent. Lyser cell, cell. In other words, the reagent that lyses the blood. Then you have another one we call fluorocell from the fluorescent. So those ones are the reagent that stairs the cells. Just look at it like that. So I'm going to take it again. Lyser cell talks about the reagent that lyse. Okay, it will lyse the whole blood. And also when you look at something like fluorocell, they are the one that will then stain the whole blood. Now let me also go further. What it means that with the Lyser cell, it will lyse the whole blood to expose the content of the blood. With the fluorocell, it can then stain whatever thing that it wants to stain, so that you can be able to measure that through fluorescence, okay, or flow cytometry. Okay, I hope you understand that. So I'm going to try to list the reagent that that are common with the Sysmex full blood count analyzer. Okay, so let's get into it. Number one, under Lyser cell, you can have something like Sulfolyser. Then you can have something like Lyser cell WNR. So when you hear the word W, it means white cell. Then NR talks about nucleated red cell. Okay, I'll take it out again. You have Sulfolyser, you have Lyser cell WNR, you have Lyser cell WDF. Again, white cells, DF means differential. Then you have Lyser cell WPC. WPC then means white progenitor cells. I'm going to get into that WPC more, but what I'm going to explain it more. But basically, with this Lyser cell WPC, this is actually when sometimes analyzer will just measure using the generic reagent, okay, like Lyser cell WDF. It can use that and give you a certain kind of result that may not be accurate. It might tell you it's a blast. It might tell you it's a typical lymphocyte and all of that. But if you doubt that, if you process that sample under WPC channel, it can then specifically tell you. It can sometimes, in most cases, if it is not a blast, then you find out that it will correct that, or maybe if it's not a typical lymphocyte, it will also correct that. I'll explain that in detail. So I've mentioned Sulfolyser, Lyser cell WDF, Lyser cell WNR, and Lyser cell WPC. Okay. Now the next other one, okay, is now Fluorocell. The same thing. Then you have Fluorocell WDF. Okay. You have Fluorocell WNR. You have Fluorocell reticulocyte, you have Fluorocell platelet, then you also have Fluorocell WPC. Okay, so now what I want you to understand is that with the Lyser cells, they can lyse the cells and expose it, but when you talk to Fluorocell, they are kind of a little bit specific because they stain a particular thing. That's why you can say when you see something like Fluorocell retics, it will just stain the retics, nothing else. When you hear something like Fluorocell platelet, it will stain the platelet. When something like Fluorocell WDF, what it will do is that it is specific to stain neutrophils, monocyte, all the differential count of the white cell count. Okay. Then if you talk about Fluorocell WNR, it is specific, especially when it has to do with something like nucleated red cells, okay, or total white cell count. So these are the kind of things that I want you to think about. Once again, when you think about in terms of the Sysmex XN full blood count, their reagents are in two forms: Lyser cell and Fluorocell. Lyser cell lyse the blood, Fluorocell stain the content of the blood. Okay. Now let us look at the functions of each of these things. So I've already generalized it. So even if you go for interview and you can't remember each of these things I'm going to say further, what you need to do then is just to tell them that the equipment using the Sysmex use something like Lyser cell and Fluorocell. Then explain what I said about the Lyser cell, explain what I've said about Fluorocell. That way you can't get it wrong. Okay. They will understand. But if you can go further for what I want to say, that will be very, very important. So now let's look at the function of Sulfolyser. What does Sulfolyser do? Sulfolyser is specifically for hemoglobin. Okay. So what happens is that now, so once the red blood cells and white cells has been lysed, okay. So then following the lysis of these white cells and red blood cells, remember that hemoglobin is on the red blood cell. The red blood cell will be treated with the Sulfolyser. Following that treatment, that will then help for the hemoglobin conversion, and then hemoglobin then will be measured. Okay. Now let me take that again. So once red blood cells and white cells have been lysed, okay. Now remember it's Sulfolyser, so it will lyse it. So once it has lysed the red cells and white cells, then the Sulfolyser will then be treated. Will then be used to treat the red blood cell, and then that can now help to expose the hemoglobin, and then hemoglobin then will be converted, and then that will be measured. That is for Sulfolyser. So when we talk about Sulfolyser, basically what it does has to do with the hemoglobin. Okay, it helps to expose the hemoglobin for it to be properly measured. Okay, now how it works is that, like I've said before, it works by cyanide free sodium lauryl sulfate. Okay, now let us look at Lyser cell WNR. Again, from the word Lyser cell, is going to lyse the blood or perforate. Sometimes they can use the word perforate. Perforate means to pierce. So once the whole blood, once the cells is pierced, that's lysing anyway. So it perforates the cell membranes. Okay, following the perforation of that very cell membrane with the Lyser cell WNR. Remember, with the WNR, you're looking at nucleated red cells. Okay, that would then lead to the hemolysis, and following that hemolysis, it can now help for the dissolution of reticulocytes and matured red blood cell. Remember that mature red blood cell does not have nucleus. Okay? Then it cannot differentially disrupt the cytoplasmic membrane of nucleated red cells and white cell. Now, because that has now differentially separated this, that can now expose the nucleated red cells. So that is how that comes in. Remember, once it exposes, then what is going to happen? Don't forget about what we talk about fluorocell. We have fluorocell WNR that can then stain the nucleated red cells. Have you seen it? So that's how that functions. But for the lysine aspect of it, the lyser cell WNR will then perforate the cell membrane, and following the perforation of the cell membrane, that will lead to the hemolysis and then leading to the dissolution of reticulocytes and also matured red blood cell. Remember that mature red blood cell does not have nucleus, but what you are looking for on this occasion is nucleated red cell. So what is going to happen? It will then differentially disrupt the cytoplasmic membrane of the nucleated red cells and the white cell. And of course, like I've said, then once that is exposed, the fluorocell can now come in to stain that. Okay, now let's move into the next one, which is the lyser cell WDF. Again, the same method, it will perforate the cell membrane, and following that very perforation of the cell membrane, then again it will expose the cell content, it will expose the content, and with the help of the fluorocell WDF, it can then stain the cells, and then that can now help. Basically, it will stain the differentials, such as something like neutrophils, lymphocyte, monocyte, and eosinophils. Okay, it will stain them. I think basophils is in another channel, so it will stain it, and that can now help the analyzer to be able to quantify this very differential. Okay, then when you have another one like lyser cell WPC, remember that I said that WPC means white progenitor cells. So I'm going to try to explain this before I explain it. So I was trying to say before that when you process a full blood count, sometimes depending on the size of the cell, it may think that this is a typical lymphocyte. It may even think that it's a blast, but it's not. So once you get such results where you are suspecting that this analyzer may have been flagging something that is not, remember that if you are working in the lab and you see a blast flag, you don't just relegate that, you don't just ignore that, you need to do something about it. And if you think this is wrong, that is where this WPC comes in. What it means that you then need to use a WPC channel to rerun the sample, okay, so that way it is more specific, okay, so to be able to correct whether that is, then if it is genuine, you will still see it, but if it is not genuine, you will no longer see that very flag, because then it will correct it. Anyway, so with the white cell progenitor cells, which is WPC, when you look at lyser cell WPC, again, the same thing, perforation of the cells, exposing the cell content, or lysing of the cells, exposing the cell content, and with the combination of the fluorocell WPC, that can now help to stain the cell content and again bring specificity to what I have already just explained to you guys. Okay, so what it means that with the WPC, it can further classify the cells. You see it now, because you think that the cell, there is some that what you think that what is flagging, once you suspect that that is not correct, then you can now use WPC channel. It can now further classify that. Once it further classifies that, you see it now, then that can now determine whether it is genuine or it is not genuine. So that is the importance of that. Okay, now that you've known about lyser cell, fluorocell WNR or WDF or fluorocell platelet or Platelet-F, whatever, fluorocell retics, what they would then be doing is to stain the specific cells, following the lysis, following the exposure of the cells, okay, by lysis of that very cell of the blood of the cell. So once that is lysed, the cells are lysed, or blood is lysed, so they will now expose the content of that very blood. Following the exposure of the content, that each of these very fluorescent can then stain specifically, and that helps the analyzer to be able to measure what is there, especially through fluorescent flow cytometry. Okay, now I'm going to go through it one after the other. When you look at fluorocell WNR, so what is going to happen is going to stain the nucleated red cell, and also it will stain the WBC, which is white blood cell. Okay, so it will stain that, and following the staining, it can then help for the analyzer to be able to quantify that. So remember, at this point of fluorocell, the cells are already exposed based on lysis or perforating of the cell membrane, so which I've already explained. Next one, you look at fluorocell WDF. Once again, what the fluorocell WDF is going to do is to stain differential cells like neutrophils, lymphocyte, monocyte, and eosinophil. It will stain it. then that will also help to be quantified through fluorescent flow cytometry. Then you have fluorescell retics, that will again stain the reticulocyte, that will then help to quantify the reticulocyte count of the patient. Then you have fluorescell platelet or Platelet-F, that will then help, of course, to stain the platelets, okay? Then that will again help to quantify the amount of the platelet. Then I have said already before that with the Platelet-F, what happened is that, you know, there are a lot of fragmented cells, okay, that is the same size with the platelet. So sometimes the analyzer might mistakenly call them platelets. So that will lead to an interference. But with the Platelet-F, it is more specific. So when you use something like fluorescent Platelet-F, it will specifically stain the RNA of the platelet and making it more specific for the analyzer to measure just the platelet. So meaning that all the interference or such as the fragmented cells which are of the same size of the platelet will not be counted. So with the Platelet-F, the platelet count is more accurate. So yeah, here you go. When you now look at the functions of the reagent in terms of the Sysmex XN full blood count analyzer, this is what you have to say. This is what it is. This is what it means. So you let me know how this video has helped you or what you think about the video. If you do have any question, of course, feel free to ask me any question and put your comment. Can I also ask you to like, subscribe, and share? Thank you very much till I come back your way again. Bye-bye.